Okay, so welcome to the third lesson or the third lab for this microarray data analysis. So what we have done over the last 2 weeks, especially, let me go recap first. First week of laboratory 1, we have a set of microarray data set, and we are able to deduce that there are 4 different categories, 4 different cancer subtypes in the brain tumor. Okay. So in the second lab, what we have done is we have looked at what genes are overexpressed in tumor or in subtype 1, that means stage 1 tumor versus the rest. In stage 2 tumor versus the rest, stage 3 tumor versus the rest, stage 4 tumor versus the rest. So by the end of week 2 or the end of the second laboratory session, you are able to deduce some form of biological meaning. So why do the brain tumor from Koo et al. Go from stage 1? How does it proceed from stage 1 to stage 2? From stage 2 to stage 3, stage 3 to stage 4. So what we are going to do today is to implement a machine learning classifier and to test this classifier based on our independent data set. So this it corresponds to the lecture 3. Essentially, apologies. Essentially, what we are trying to do is, since we have expressed genes for genes that is upregulated in stage 1, stage 2, stage 3, stage 4, can we build a machine learning classifier that when I have a new patient comes in, I do a microarray analysis, can it actually tell me which stage is this patient in? Okay, so this is quite useful, but before we do that, we have to test our machine learning classifier. Okay, and remember, we cannot test it based on the training data that we give. So the training data come from Koo et al. We have to use a totally different data set to test it. Otherwise, it will be testing itself. Okay, so let's get started. The first thing is to load up the libraries. Okay, assuming that all of you have installed already from the last 2 sessions. So let's go and run the libraries. Okay. Yeah, my clock is ringing. So loading up the libraries. Okay, if there's no errors, users bioconductor, okay, everything looks fine. So remember, we have to change our working directory, okay, because now my working directory is totally different. Okay, so now I got to change my working directory. Go to set working directory, choose, and here what I have to go is my practical session and my practical tree. Okay, so now I have my working directory, and then what I want to load up is basically everything. Okay, so let me just load up all my data. Just load up one at a time. End of day 2. This is my load up every piece of information up there. Okay, remove data set, test data. This is also test data. Okay. So let's load up every single piece. We have loaded up quite a lot of data. Okay. Now then we can start doing our work. So we have loaded up all the data here already. We will not need to do it anymore. So let's look at what the groups that we have. Remember, we use NMF to look at the groups, and we actually call them as a metagenes. Okay, so metagene values by convention we call it typically H, and then we remove, remember, the last 2 negative silhouettes. Okay, then we do a coefficient map to see what happens. So this is what we have done in week 1, or rather, this is what we have done in the laboratory 1. So let's run it. Okay. So this is what we have, remember? So we have the silhouette, we have the plots. So what we have done is to group all my samples into stage 1, stage 2, stage 3, stage 4. In fact, this is stage 1, stage 2, stage 3, stage 4. Okay, so this is what we've seen in the first lab session. So nothing very much, just a kind of recap. Okay. Then after that, we realize that there's actually a fair bit of overlap. Okay, for example, week 2. Okay, a fair bit of overlap of the genes. Remember week 2, our Venn diagram showed that there's a fair bit of overlaps. So what we are trying to look for is find those genes that is correlated and anti-correlated. Okay, anti-correlated simply means, okay, when we say correlate means positive correlation. Anti-correlate means negative correlation. Okay, that's what anti-correlate means. Some people use correlate, some people use anti-correlate. Some people say that it's positive relationship, negative positive correlation versus negative correlation. Okay, so it depends on which are the terms that you want to use. So the very first step is to calculate our correlation. Okay, so our correlation, this is our correlation function. So let's put it in. Okay, it takes a lot of effort to learn how to program in R, I have to say. So I'm not expert in R myself, so I can just use R. Okay. So we have the function in already. So the function is called COR, core function. Then we can use it, start using it. We want to use it, which genes are correlate and anti-correlate, positive correlation and anti-correlation. Okay. So what we want is to have a vector and then to put the correlation into it. Okay. We can do that for first, second, third, and fourth meta gene. Okay. So let's run everything at one go. Okay. Remember, meta genes are actually hypothetical genes, basically correlated in functions. So let's run that. So now we have 4 meta genes, okay, H1 to H4. And what we want to do is to investigate, do the meta gene 1 correlates well with the rest. Okay, so let's run this. So we want to plot meta gene 1, the most correlating probes to meta gene 1. Okay, so there's a lot of functions here. Let's see what we get. Okay, we want to print out the R² value actually. Okay, so let's look at this plot by itself. Now, meta gene 1, let me just, hello, wake up, does it even wake up? Okay, doesn't seem to like me very much. Okay, but anyway, let's look at this diagram here, this small diagram. Let me just zoom again. Yeah, now it looks better. So now I have this meta gene. Okay, meta gene 1 is actually this probe, the most, the gene that the probe that is most correlated with meta gene 1. So the R² value is about 0.7. And can we actually see that it differentiates the 4 groups quite well? Okay, we see that this what you call green group seems to differentiate itself quite well with the rest, but the rest of the 3 groups or the rest of the 3 subtypes seems to be a kind of uneasy, not able to classify very well. So using this probe alone, we can actually separate based on this green, maybe one of the stages of tumor versus the rest. So this is actually quite good. Okay. So let's continue. Based on this information, we can look at the most anti-correlating probe to. So we'll look at the positive correlation. Why not we look at the negative correlation? Okay. So negative correlation, let's plot it out. Okay. So you see what actually happens here is you plot the positive correlation with negative correlation. Okay. So negative correlation, we should see the line going downwards. So do we get that? Okay. Yep, we see that. And that means this probe is most anti-correlating. That means negative correlation with meta gene 1. And what is this? The correlation is actually R² value is 0.85, which is quite decent. Okay. Similarly, it seems to separate this green subtype with the rest. Okay, so we kind of give us some very interesting idea. It seems that one of the cancer subtypes may be neatly separated from the rest using just these 2 probes. That means we can look at the expression of these 2 probes. Okay, in the microarray data. or rather the intensity of these probes, these two probes. So this can be quite interesting. That gives you some idea. Of course, we can actually then find out what is the identity of this gene. Okay, this probe actually is to measure what gene. So that can give us a lot of biological understanding as well. Okay, just by looking at all this information. Okay. Now you can actually do more if you want to. And what we want to do is, can we actually just correlate everything together, relationship between all these probes. Okay. So what we want is to do a Venn diagram of everything. Okay. So we look at that. Okay. Now, if you look at the Venn diagram here, okay, we have no overlaps. So we can find that there are 40 probes that is significant to group 1, 4, 2, 3, and 4. So there are no overlaps. These 4 meta-genes seems to be pretty useful. This 4 set of meta-genes seems to be pretty useful. Okay, and we have you do not have meta-genes that is correlated or anti-correlated with meta-gene 1 that is also correlated with meta-gene 2 and so on. So this can be quite useful. Why not we look at these 160 probes and see what happens? Can we actually use this? So remember our microarray data, each sample, each clinical sample, each array has 1,500 probes, but we can actually narrow down to 160, which seems to able to differentiate our cancer subtypes well enough. At least it seems to be a case. So let's take this further and see where can we go from here? Does it eventually break down? So let's continue. So there is actually no overlapping of the probes to each meta-gene. So why not we take this information and we actually put them into a heat map to actually see what happens. Okay. So this set of genes, let's put them in heat map first and see what the results we get. Okay. Now, if we actually plot it with a heat map, look at this. So this is, does it look pretty much like what we have, our separating line? So this may be group 1 or one group of one stage of tumor. The red one is one stage of tumor, the yellow one is one stage of tumor, the green one is another stage of tumor. Of course, there's one that is seems to be error, but on the whole, it seems okay. You see this green line, this green sample, seems to be the kind of the odd one out, right? But the rest seems to be quite okay, especially between the tumor subtype for this represented by this blue and versus red. You can see that from here, these samples, they are distinctly different from the red samples. So it could give us a lot of information already. Okay. So let's move on from this step. And why don't we just continue it and do a principal components analysis with just these 160 probes. Okay, because why? How do we get that? We have the joint values already. Okay, and using the joint values, we actually pull out this filter.tree. So this is just the filter.tree data is just 160 probes from our 1,500 probes. Right? So can we just use these 160 probes to actually do? So we put 160 probes in, data in. It's actually a reduced data set. Okay, originally we have 62 samples. Over each sample have 1,500 probes. Remember, we removed 2 samples that give us a negative silhouette. Okay. So we end up with 60 samples, each sample 1,500 probes. But now we have joined the correlations up, and we only have 60 samples, each sample have 160 probes. Okay. So why not we do a principal component analysis? And we call this as filter.tree.pca. Okay. Then we kind of take the first 4 principal components and just plot it out and see what happens. Okay, we have done this in our first practical as well. So let's run it. Okay. Now, does this 4 actually looks kind of well? Is it? Can we see that there are some separation, some neat separation? Okay, the blue and the red seems to be well separated. The blue and the red seems to be quite okay, right? The only problem is the yellow and the green. Okay, in some cases seems to be well separated. In some cases doesn't seem to be. Okay, for example, here there's a bit of overlap. This kind of separates quite well. This all right, a little bit of overlap. This separates decently well. In fact, you can just draw a line across here. Separates well enough. Okay, for this you can just draw a diagonal line here. Separates well. Okay. So this tells you that, okay, seems to be, okay, we are getting somewhere. We are getting a way, we are getting some sense that these 4 tumor subtypes may be separable. This is very important. If everything is jumbled up in a big mess, then it tells us that maybe things are not separable. Now it tells us that things may be separable. Why not we try to build a classifier and see what happens? Because at the end of it, when we test our classifier, it will really tell us, are we doing a good job? Okay. So let's continue moving on at this stage. So what we want to do is, can we take these 160 probes to build a machine learning classifier and then test it on an independent data set? We call this independent data set as TS. Okay, remember our TS file? We have this TS file here. Okay, so this is our independent data set, which we have actually bring it up into the environment already. Okay, so no problem with that. Let's continue. So SVM, the support vector machine, we are going to build it. And we use a linear support vector machine in the first place. Okay, remember, linear support vector machine has only one parameter, cost, to optimize. Okay, so in order to do that, we just do a grid search. Okay, so we just try a range of values. Okay, 2 times 10 to the power minus 5, 2 times that means the cost. 2 times 10 to the power minus 5, minus 4, minus 3, minus 2, minus 1, 0, 1, 2, 3, 4, 5. Okay, and for each one, we do a 10-fold cross validation. Quite cool, right? So we can actually do that. And how do we do that? Let me just settle this, make it clearer. Okay, so we use this thing known as, we want to train. Okay, the data is called train, and we use it based on our expression, our filtered.tree. Okay, we want to train based on the x value, the data that we give it is the filter expression.tree. The answer is actually in our group 2. Remember, our group 2 give us all the answers in the first place. Let's just look at what is in group 2. Okay, group. Okay, remember this group 2? Tell us a fair bit of each sample is group 2, this variable. Every microarray data, every microarray sample belongs to group 1, group 2, group 3, or group 4. So that is our answer. We have the training set and the answer. Okay. We test using an SVM, and we want to tune the SVM based on what? Based on a tolerance, and we use a classifier, it's a linear kernel. Seed, remember this is the random seed that we produce, give it, and what range do we want? Okay. The range that we want is, let me just get this out clearer. The range that we want is from 2 to the power of minus 5 all the way to 2 to the power of 5. Okay. And we want to do, how do we test it? We want to do a 10-fold cross validation. So tune control is using cross, and cross 10 means 10-fold cross validation. Okay, this will take some time because what you are doing is you are doing minus 5, minus 4, minus 3, minus 2, minus 1, 0, 1, 2, 3, 4, 5. Actually 11 support vector machines and test each one independently. 11 support vector machine, 10-fold cross validation. You are actually running it 11 times 10. So it's 110 machines. So it's actually quite a lot. So let's do it. If your computer is actually a little bit old, when I'm talking about old means maybe a couple of years, please go for a break. Okay. So let me just set up. Otherwise, it probably will take a few minutes. Okay. So now it's finished already on my computer. It's fast. Okay. Lucky to have a fast computer. Now, the first time when I actually run this session, this step alone takes half an hour. So don't be surprised. Okay. So now my classifier tune, what I want to find out is which is the cost that gives me the best result. Okay. So I pick now classifier. Within classifier, the best cost, the best model, the best model, and then what is the cost? Okay. I put it out and see what happens, and actually call for the cost. So it tells me that the best cost is 0.03125. Okay. So what you're saying is 2 to the power of, okay, so 2 to the power of n equals to 0.03125. Okay, so what is n? Okay. Let's take a calculator and just run it and see what happens. Okay, so essentially n will be equals to, I can do a log, I can do a ln even, 0.03125 divided by ln 2. Okay. Let me try on my calculator. 0.03125 divided by ln of 2. Okay. And what I have is the best cost is -5. So it's within the range. Okay. So this is like a gross estimation. Next is we want to tune it better. Okay. So since the best estimate is 0.03125, why not we tune it better? We tune from 0.01 to 0.06. Okay, 0.01 to 0.06, and we increase the interval by 0.002. Okay, so what does this really mean? It means that what we are tuning it for is actually from 0.01, 0.0102, 0.0104, and so on, all the way to 0.0508, and last one is 0.06. Okay, so this is what happens. If you look at the okay, let me just change the view on this. Let me change to a web view. Okay, this may be a bit clearer for you. Okay, take zoom. Change to web view. This will be a bit clearer. So originally we have cost is 2 times 10 to the power of 2 times 10 to the power of -5 to 2 to the power of 5. So here what we want is everything remains the same except this line here. Okay, let me just get back. Except this line here. Okay, so the cost is actually now a list. It's still a list. The cost is equals to something, right? But it's a sequence from 0.01 to 0.06, incremental interval of 0.02. And then we still use our tenfold cross validation. Okay. So what does support vector machine, what is useful of support vector machine is you can actually do it anything. All you have to do is change the X and Y data. Remember, the Y is the answer. The X is the data that you want to train it with. Okay, it is very similar to what you do your regression analysis. Y equals to sum B1 of X1 plus B2 of X2 plus Bn of Xn must be 0, correct? So this is all the x values, which is actually, in this case, our 160 probes. You want to classify based on the answer 1, 2, 3, or 4. Okay. Y is the groups. So let's run this. This will take even longer because, as you realize, 0.01 to 0.2, 0.02. Okay, so sorry, this will be here 0.02. Otherwise, it'll take even longer. So okay. So you see, 1, 12, 14, 16. So it's 10, 12, 14, 16, 18 until you get to 60, essentially. Okay, so this we have 10. I'm taking the last two is 10, 12, 14, 16, 18, 20, 22, 24, all the way until 59, 60. Correct. Okay. So let's run this step and we'll see what happens. Let me just get this out of the way. Okay. This will really take a little bit of time. I don't know, maybe a few more minutes. Well, okay, it's actually less than a minute. Sorry again. Once you have a better, faster computer, you kind of lose your coffee break. And I thought that while waiting for this, I could go and eat my cornetto ice cream, but apparently I can't. All right, so let's continue then. We do the same. We find what is the, hey, we use the same variable because really we do not need this anymore. Okay, we use the same variable and we find out that what is the best cost now. Okay, so this is the same. Okay, so the best cost is 0.01 in this case. Okay, so it does depend on a machine differently. Okay, so over time is a bit better. Now, if you go from 0.01, maybe you can do it a bit better and so on. Okay, that's up to you. Okay, if it's, let's say, normally if I were to do it, okay, if let's say for here it hits 0.06, okay, or rather hit 0.01. This is my lowest end. I may go all the way to 0.0 to begin with. Maybe that will work. So let's maybe, just for the sake of it, why not we try? We try this. If I go from 0 to 0 all the way to 0.01, and actually run it and see what happens. Is 0.01 the best cost? Okay, so it has to be more than 0. So I have to be 0.0, I don't know, give it a 0.02 then. Okay. Oops. 0.01. Okay, so this doesn't work. Let me just go back. So I have to give it 0. Of origin is 0.0. This is what I'm trying to do, 0.002. Okay, so maybe let's run this step. Okay, cost is 0.002. So it's a bit strange, but doesn't matter. Let's just take the best cost as 0.01. And we build a classifier and see what happens. So you want to build a classifier, this is quite fast. We actually change the cost to the original cost. This is what you can, let me just run this and I'll show you exactly what happens here. So rather than copying it out, I can actually use a variable, this cost equals to cost. Really, this cost, what I'm trying to do is change it to 0.01, which I found it up here. Okay, make sense? So I'm just going to build a classifier in this. Just this called my classifier. So a support vector machine classifier is really just an algorithm, and this will be very fast because there's only one. Okay, so it's finished. Build the classifier already. Then what we want to do is we want to predict. Remember, in our lecture we talked about confusion matrix. Can it predict well? So let's run a table to check whether the predicted value from the classifier versus our original data, the data that we give it. Basically, you have the answer from the classifier check with the model answer. Okay, does it work well? Okay, now it seems to be marvelously perfect. See, if it calls group 1, it is group 1, and group 2, group 3, and so on. The others is 0. Actually, this works marvelously well. And obviously, when I calculate the Cohen's kappa, it's going to be tip-top condition. Okay, you don't even need to see anymore. It's obviously going to be 1, right? Because there's no error. The accuracy is 100%. Which is, I don't know, I tend to find 100% accuracy a little bit iffy. But assuming, let's go with it and see what can we do. So now, the classifier actually performs perfectly. That's what we expect. But this may be due to overfitting. Okay, but then you think, how can overfitting really run well in a tenfold cross-validation environment? It should not. It should not. Okay, so hopefully our tenfold cross-validation kind of help us weed out overfitting. Okay. So what we want is, why the last part, after you do all this work, you cannot say that your classifier works. You need to test from a different data set. So this data set comes from another paper, which is a Nature paper. Let me just see whether can I find this paper. No, distinct group from blastoma. Let me just see. Okay, Nature 2012. Let me see if I can find. Okay, good. I can find this paper. Okay, so you go to read this paper. Okay, this is the PubMed ID number. Remember how to use PubMed ID. Okay, you can go and read and actually see what does it say. So we have a data set that can be quite useful now, and we want to see, can our classifier classify this data set? Okay, because why? This data set is also 4 groups. Interesting, right? And it's medulloblastoma, which is actually the same as what we expect, okay, from our Koo et al. Data. So let's run this and actually retest it out. First, we want to look at the heat map, whether does our classification works well enough. Okay, so let's run the heat map on the test data first, the test set, which is this Nature paper. Let's run it. Because if it runs everything well, then we should expect something to look like that. Okay, the 2 studies should be somewhat similar. Makes sense, right? So let's run it. Since we have the data already, TS is already in our environment. See? Now, you look at this, and we go back. We look at this. We can see some similarity somehow. Okay, may not be perfect, but we can see that, okay, there are 4 big groups. So why not we take this Nature paper's data set and test it using our classifier? Because why? Our classifier is built using Koo et al.'s data set. This Nature data set has not been seen by the classifier. So this gives us a real representative view. Okay, makes sense, right? So this is actually what we call as the moment of truth. Now, we want to get our classifier to predict on the new data set. Use this predict function, and then we get the output is on called test predicts. Okay, so let's run this and see what happens. Okay, it will show nothing because we are not printing out anything yet. Then we want to actually supply their calls. Their calls is based on this Nature paper, which is, let me go find the name again. we call it Robinson et al. So the first one used to build a classifier is Koo et al. So this is Robinson et al. So Robinson et al. Do have in their paper, obviously, they kind of, this is their calls. So based on this is Wnt, based on this is SSH, and so on and so forth. So their calls. So we supply their calls, and then we map it up onto their data set. Okay. So we actually put their calls in, and we kind of just slam the two things together. Okay. And then to look at their calls to see whether is everything looking well. Okay. So these are their calls. So apparently everything is labeled properly. You have Wnt, WNT, that's called Wnt, which is actually wingless pathway. SSH is a sonic hedgehog pathway. G3, G4, and then you have a few that is SSH outlier, U, unclassified. Okay, a few of them. So now, the moment of truth is we want to map it out on our predicted values, predicted groups, based on their calls. This is quite cool, because we want to see whether is, let's say, all their groups. That means every sample that they have as G4, does it fall into one subtype or one stage of tumor that we have? If it falls everything into one stage of tumor, then it's actually looking pretty well. Correct? So let's run this. We table it out, and this is usually before I run this step, to make sure my work is doing well, I kind of, for lack of a better phrase, I'll say a prayer first. Then I just type it. Okay, this is their calls. Okay, this is Robinson et al. This is test predicted. Stage 1, say group 1, group 2, group 3, group 4. Stage 1, stage 2, stage 3, stage 4, or something like that. Does it actually look well? If we remove this SSH outlier and unclassified, it seems to be really well, except G4. The G4 is separated between our group 1 and group 3. But their G3, 16 of their calls that they classify as G3 meets our group 3. All 10 that they classify as SSH also meets our group 4. The 8 that they call as Wnt meets our group 2. It's almost nearly as good as you can get, except these 3 that's mislabeled or misclassified to be exact. Okay. So why not we actually remove the SSH and the U category? We remove category 4 and 5. So this is category 1, category 2, category 3, category 4, category 5. So we remove 4 and 5. We remove SSH outlier and unclassified. So we just remove them. Okay. So how does it look? We expect it to look nearly perfect. Okay. See, it's nearly perfect. What they classify as G3, we classify as 3, group 3. What they classify as G4, mostly we classify as group 1. What they classify as sonic hedgehog, SSH, we classify as group 4. What they classify as Wnt, we classify as group 8. We classify as group 2. See, there's actually no error. Marvelous. Marvelous. Okay. So why not we relabel them? We say that, okay, their call is 3, then every time our predicted value is 1, group 1, we call it as G4 and so on. Okay, so this is exactly what it does. Okay. So if the test predict is 1, we call it as G4. If the test predict is 2, we call it as Wnt. If the test predict is 3, we call it G3. Test predict is 4, we call it SSH. Okay. Basically, relabel our predicted results. Okay. And everything else that falls out of the category, we call it as NA. Okay. So we just do this. It's just a relabeling step, nothing very much. Okay. And once we do that, we can actually look at it all over again. Run this step, we run the table. See, when we predict group 3, we call it as G3. So this is where the 16 come from. When we predict as group 1, we call it G4. When we predict as group 4, we call it SSH. When we predict as group 2, we call it WINT. So there is, it's not as perfect as the first one, right? There's some error. But you realize that no, the numbers actually still look very decent. So why not we calculate the Cohen's kappa? Okay. It will give us a good result. Okay, because the error is very small. That means our accuracy, our machine learning classifier accuracy is actually 93.66%, which is very high. I would say very, very high. Okay. So what we do is, can we label, we put our results with their results, and then we kind of plot a heat map all together. Okay. So see, this is what happens. We plot a heat map all together. The bottom row, because of we know that there's a gray, is their calls. Basically, is Robinson et al.'s data. The name predict is the first one. Where are the errors? We can actually see where are the errors. Okay, so the errors are here. These are the errors, and then this error. Then we have a few errors that seems to be here. Okay, so because the gray ones, the SSH and unclassified, we throw them away. So our 3 errors is this 3. This yellow, and this yellow. So that matches these 3 errors here. Okay, so it's actually not too bad. Okay, then what we want to do next is we do a PCA plot of what their assignment versus our assignment. Okay, so we have we plot the assigned groups. Basically, this is more of a, we already kind of certain up to this point that things are doing well. In fact, this is the most important table. Things are doing well. This is also a very important diagram. It's actually publishable diagram. Okay? Things are doing well. So the rest is just redoing our principal components analysis to actually see what happens. Okay, there is a little bit of error here. Okay, so don't bother about that. But all this is kind of what we kind of need to do. Okay, essentially, just do a few more principal components analysis. Am I lost on somewhere? Okay, never mind. Just don't bother about it. Okay. Then what we have is, okay, is to run a set of silhouette plots again to actually see what has happened. Okay, let's run our silhouette plots. Okay. Okay. So this is our predicted cost so far. It seems to be doing decently well. Okay. And this set of data is from Robinson et al. We have some errors here. That's fine. Okay. So this is more of trying to pull out more data. But as I say, up to here is already the best thing that you can have. This best thing you can have already. So the rest, I will leave it up to you to do. There is nothing really very much that we need to do extra. Okay. And that's all. Do you think our classifier actually works pretty well? What I'm suspecting is I have kind of, yeah, basically see, I've duplicated my stuff. Okay, so this part will be dumped away. Okay. So we have actually finished off everything. Our classifier actually works decently well, but it wasn't perfect. We can make it better. So we'll think about how do we can make it better. Okay. Okay, sorry, I was just out for a little while. My nice brother, my good brother, has just bought me some stingray nasi biryani for my lunch tomorrow. So I go and get it right. You can't blame me. Okay, so all of you have to wait. Okay, so anyway, let's continue. So essentially, our data is actually doing pretty well. Okay, our data is actually doing well enough. We have nearly 94%. But if let's say our data is our result is maybe like 70%, then how can we make it better? There are some tricks or some ways you can do it. Maybe you can actually do more on the probe selection. We only select 160 probes. Okay? Since 160 probes are working well, can I just kind of for tumor subtype 160? Can I kind of reduce it to maybe 80? Will it be even will the result actually suffer too much? Or if my results, my accuracy is poor, can I increase it? What is my better probe selection? Okay. Our support linear vector machine. Our linear vector machine is actually a simple SVM, a linear vector machine. So it's actually the base model. Okay. Can we make it better? For example, remember we can use a non-linear support vector machine. And then what we also have is we have now one SVM. The result is 4 output. 4 result output. Okay. So if my my result is poor, can I actually have, let's say, SVM A that separates maybe stage 1 and 2 versus stage 3 and 4 tumor. Then I have from then on as SVM, the second SVM will take the result. If let's say it tells me at stage 1, okay, take the result and go to the second SVM and then use classify it to 1 versus 2. So it's a stepwise classification. Then of course your SVM, the third SVM, what am I doing? The third SVM can actually classify between if the SVM first SVM actually tells me it's maybe stage 3 and 4, I can throw in the third SVM and classify stage 3 versus 4. You know, there are many ways of how you can play around with this. It is only usable when, or it's only effective when, let's say, if our accuracy is low. But we can also use the same method. How much do you want to increase? We are actually doing 93.6, 93.7%. And really, misclassification is only 3 of them. Of course, you may argue that clinically, if you actually, I'll say, diagnose a person that is supposed to be stage 4 tumor, and you tell him or her that it's stage 3 tumor, maybe you will lose some treatment modalities, some treatment options. But we have to remember that all these is a first step. You have to go and look at the data. This microarray, you have to do more analysis before you actually have an actual treatment. So how much more accuracy, how much more work do I want to increase accuracy? Maybe do I reduce this from 3 to 2? Can I reduce this from 3 to 1? What kind of added benefit will it have as a first stage classifier? So this you have to ask yourself. Okay, how much more work to get to where you are? It's almost like saying that if for maths paper, you score 94 marks. How much extra work you want to put in to score 98 marks? It's up to you to think about it. Okay. So all in all, we have finished all the 3 lab session. Just to recap, the first lab session, laboratory 1, you are given Koo et al.'s data set. You do not know, Koo et al. Say that there are 5 different subgroups, maybe 5 different stages. We disagree. Our data, our analysis tells us that it may be only 4, and we can't go with 4. Okay. Again, if we go with 5, what is the kind of things that we can have? We do not know. Okay. Using this, the second lab session 2, we deduce, okay, stage 1, what is the genes that is expressed, significantly expressed over the other stages. Stage 2 over other stages, stage 3 over other stages, stage 4 over other stages. Deduce the biological functions using gene ontology enrichment analysis. So this gives you some clinical, some biological basis why the tumor go from stage 1 to stage 2, stage 2 to stage 3, stage 3 to stage 4. Now, if you are quite sure that there are only 4 stages, the third laboratory session, which is now, we build a classifier, okay, using Koo et al.'s data set, and we use another published paper, a totally different data set, Robinson et al., to test, and our accuracy is nearly 94%. Okay. So how are you going to do all this? Let's say a new set of data. This whole set of exercise, as I say, is almost like a recipe. There's only really 3 pieces of data that you have to reassemble yourself. What is the first one? The first one is this filter.expression, which is really Koo et al.'s data set. Okay. If you remember, filter.expression, you have your microarray, sample 1, sample 2, and each column is 1 microarray. Each row is a probe value. Okay. So columns, 1 sample, columns, 1 sample, row is 1 probe. This we have it quite easily. Our testing data set, okay, Robinson et al. TS, we look at it. Same thing. So TS and filter expression is same thing. Column, 1 sample, 1 column, 1 sample. Row is probe. After that, what is TS.PD? PD is their calls. So their calls is actually, now you reverse it from the columns, you actually swap it, transpose it into rows. So each microarray is 1 row. It's almost a description. Okay, the title, the subgroup, and this is the subgroup characteristics that we are looking for. Okay, the rest of it doesn't make any difference. Okay, age, gender, all these different other stuff. Okay, so if you can example your data this way, these 3 pieces of data, you can actually repeat this whole experiment with, or rather repeat this whole 3 exercises with almost any form of data set. All you have to decide is how many groups in the original data you have. So I leave it up to you to go and think how you can apply this elsewhere. Okay, and I do encourage you to keep these 3 sets of notes with the data set so that you know how to actually analyze your own data in future. Okay, with that, good luck for your exam and good luck for future work. Have fun. Bye-bye.